We had some exciting sessions in between time, and let's see, Alicia has told me that she's going to post into the chat window some information on where everyone can find the narratives that were used in the hands-on sessions by the student teams. So everyone who wants to go through those same narratives that the student teams went through will have access to that. So watch the chat for that information on where to find those. And so this afternoon we have another presentation, and then we'll have another short hands-on session that'll be private. So we'll be off of this Zoom for a little while, and then we'll be back again for our final session. And so we have some exciting things planned for you this afternoon. And the first presentation today is going to be given by Mike Shaffer and Kayla Borton, who are from Kelly Wrighton's group at Colorado State University. They're going to be presenting on the topic of inferring biogeochemical function from genomes. So with that, I'll hand it over to Kayla and Mike.
Great, thanks, Tim. Okay. And everyone's seeing my slides?
Yep, got it.
Great. Thanks, everyone. So like Tim said, today we're going to be talking about going from bins to metabolic potential. It's going to start off with me presenting some slides. Then we're going to switch over to Kayla about halfway through. So I'd like to start off by saying congratulations, you have bins. For all of those that were working in our small groups, they got to go through a narrative where we created those, and everyone else was hopefully able to hear the lecture this morning where Kelly talked through how you can go from reads to bins. Some things you'll need to remember as we go through the talk this afternoon is first to remember that ideally each bin represents a genome from your community. So as we learned this morning, we have reads that we use to assemble into contigs. Then we bin those contigs. So each bin is a list of contigs. And together we call those metagenome assembled genomes, because hopefully these contigs represent a single genome from our community, a single partial genome from our community, hopefully complete as possible. And from that genome, we can learn a lot about an organism. So this figure here on the right is from one of Kelly's papers from 2014, where each of these little cartoons is representative of a bin that she was able to pull out of an analysis. So it's showing the metabolisms that each bin are able to do. And that's what we're going to work towards being able to do today, going from these bins to understanding the metabolism that each bin is potentially capable of. So our goals for the afternoon, first is to do a quality control check to ask, are our bins high quality? As Kelly briefly mentioned this morning, we need to be able to understand how good our bins are, and should we be able to trust them? Is this really representing a biological unit within our community? Then we want to be able to assign taxonomy. So this is asking, what is this organism? So what taxonomic group is it from, and what can this tell us about the organism? Then we're going to want to annotate with function. That's asking, what can this organism do? What functions are it capable of doing, and how will this impact the community? Which leads us to question 4, which is our topic 4, which is using the functional annotation to understand potential metabolism. So how does organism act as a member of a larger community? So this is just going to introduce a little cartoon that I'm going to be using for the next few slides, and answering the question of what is a genome. So hopefully we remember that a genome is the sequence of DNA in an organism. So in our example here, you can see that we have a bacterial cell as represented by the blue box. And in that we have a darker blue circle, which has some bars on it. So that circle represents our circular bacterial genome, a genome that really exists in the biological sample that we took. And on that genome, there's a bunch of different bars of different colors. And those bars represent genes that are present in the genome. So for example, here, our little genome would have 7 different genes. So first, we're going to talk about quality assessment. So how can we assess that the bins or mags that we're able to pull out from our sample are of high quality and represent a real biological unit? So we know that bins are partially complete genomes. And so you can see our circular genome up here on the right again, and that there's all these different colors on it. But with this blue bar, there's only one of those. So that's a single copy number gene. And for example, we could have this bin. So say this is the bin that we're actually able to make by assembling our reads into contigs and then binning our contigs into a bin or mag. And you can see here that this is somewhat representative of the genome that was really there in the community. For example, you can see that there's this, in this bottom bar, there's this pattern of orange, yellow, orange. So that might be this section on the upper left part of our genome. And then we have this other section with green, yellow, blue. That might be this section of our genome. And so what we want to know is this bin that we made, how representative of this, or do we think this is a real thing in our community? And to do this, we want to be able to measure two different things. First is completeness. So that's measuring how complete is this genome. Of the genomic content of this organism, how much are we actually representing with this bin? And then second is contamination. Contamination is asking, are there parts of other genomes that are present in this bin? So how purely is this bin representing only one organism? And both of these are measured by looking for single copy number genes, like that blue gene present here on our real genome. What are single copy number genes? Well, it's in the name. It's genes that only occur once within a genome. And as we can see in this figure here, where our x-axis is genome size, so as we move along to the right, genomes are getting larger and larger, and the y-axis is the number of genes in a category. And if we look at, for example, the purple category, which is transcription, we can see that as our genomes get larger, we see more genes that are involved in transcription. But if we look at the red genes, which is a selected group of 35 marker genes, no matter how big the genomes get, we still have the same number of marker genes. This means that these seem to be present in the genes that make up that group of 35 seem to be only present once in each of these genomes, no matter how big they are. That means that we can look for these marker genes and use that as a measure of how complete our genomes are. So we can use these single copy number genes to understand completeness and contamination. So if we look at our example bin 1 here in the upper right, we can see that this bin is somewhat incomplete because it's missing the blue single copy number gene. We know that if this bin was 100% complete, it would include the blue gene, but it doesn't, so therefore it's incomplete. Then we can also look at our bin 2, example here in the bottom right. In this bin, we see that there are 2 copies of the blue gene. This means this bin is contaminated. We know based on that plot from the previous slide that each genome should only have 1 copy of the blue gene, and therefore this genome is contaminated. To assess quality or completeness and contamination, we use a tool called CheckM. CheckM is available in K-Base, and it's what we used in the narratives that the groups went through today, and everyone will be able to see soon. And CheckM is more advanced than just looking for that simple set of 35 universal marker genes. It looks for marker genes that are specific to clades. So for example, the team behind CheckM looked at all of proteobacteria and found genes that are present in single copy numbers only in proteobacteria. That creates a bigger list than if we just look for 35 genes which are present across all bacteria, and allows us to have a better measurement of quality and contamination based on an assignment of the bin to a specific taxonomic lineage. So CheckM does that identification of the lineage and then looks for single copy number genes that are specific. And then with that, it reports the completeness and contamination. And on the right here is an output of what CheckM looks like when you run CheckM in K-Base. If you look at the bin 79, the top of those 2 bars, you see that there's a lot of little lines on there. That means there's a large number of marker genes that are associated with the lineage that bin 79 has been assigned to. And the fact that almost all of them are green means this bin is very complete. And the fact that there's not much red means there's little contamination. Whereas if we look at bin 2, we can see that there's a smaller number of bars, which means there's a smaller number of marker genes associated with that lineage. And that a lot of the bars are red, which means there's a very high amount of contamination. So if we were doing our analysis, we might want to move forward with bin 79, but not move forward with bin 2. But then how do we decide which bins move forward? How do we determine what is a good quality genome? So luckily there's a group which developed guidelines for this, which called the minimum information about metagenome assembled genome, or MEMAG. And they defined criteria for what it means to be a high quality, medium quality, or low quality genome. We consider a high quality genome to be one where the completeness is greater than 90%. So it's to the right of 90 on the x-axis. And the contamination to be less than 5%. So above 5, which in case 0 is at the top of the y-axis in this plot, means that it's low contamination, below 5%. And so therefore it's only the bins, which are represented by dots, which are in the upper right-hand corner with the green background of this plot, that are high quality. As well, we also define that medium quality as greater than 50%, so to the right of 50% on the x-axis, and less than 10% or above 10 on the y-axis. And so everything in yellow will be our medium quality genomes. And generally, when we're doing a MAG-based analysis, we will use all medium and high quality genomes when going into further analysis and trying to understand the metabolisms that each bin is capable of. Next, we want to assign taxonomy. So now that we have our bins, we know that they're good quality, and we think they represent only a single organism. We want to know what taxonomy these bins represent. So this is asking the question of who is there, what actual taxonomic units are present in our genome database. So we do that by identifying the genes that are present in each genome and then comparing that to a database of genomes that have known taxonomy. For example, many of you probably know about the NCBI genomes database that has a set of genomes and taxonomy assigned with each of them. So we could compare our genomes to that database and say, if the genes in our MAG are very similar to the genes of a genome in that database, then they are likely the same taxa. To do this in KBase, we use a tool called GTDBTK, which stands for Genome Taxonomy Database Toolkit, which will assign taxonomy based on genome sequences, especially from MAGs. GTDBTK will compare your bins to a massive database, which is GTDB, which contains around 25,000 different genomes from bacteria and archaea, including from other MAGs, and assign taxonomy based on that comparison. And an important thing that GTDBTK does is that it will assign partial taxa strings, so it won't give you your full taxonomy down to the species level when no very close relatives are found. For example, you can look at the 3 trees that are included at the bottom here. In tree A, you can see that there's the phylum Proteobacteria and the phylum Actinobacteria, which are clustering together within this branch. But outside of that group is our query sequence. So that's going to be our MAG of interest. And because it's clustering away from all the other known phyla, we'd say this is a novel phylum and only assign the domain bacteria to this organism. With example B, we see that our query is clustering within other classes within the phylum Actinobacteria. So that would allow us to say this allow us to assign the domain bacteria and phylum Actinobacteria, but we would say it's a novel class. And finally, in example C, we see that our query is clustering with Escherichia, but away from Citrobacter. And therefore, this is a novel species within the Escherichia genus. So now we have bins, which we are sure are of good quality, and also that have been assigned taxonomy. But what we really want to do is build those genome cartoons, as we showed in the initial slide. We want to be able to understand the metabolism that each genome is capable of. So we can do this with a tool that we've developed within the Wrighton Lab called DRAM, which stands for Distilled and Refined Annotations of Metabolism. DRAM can be broken up into 2 steps, which are each further broken up into 2 steps. The first part of DRAM is called annotation. This is a similar pipeline to what most other annotators use, such as Proca or MetaErg. It first starts by gene calling, which is taking that genome sequence, so that collection of scaffolds, and then for each of those, finding the genes that are present on each scaffold. Then it takes those gene sequences and searches them against a variety of databases to assign function. In doing this, we can understand all the potential roles of all the genes in each genome. But what really makes DRAM special and separates it from other annotation tools is the distillation process. The first part of distillation is the functional classification of genomes. So we'll go through all the annotations from all of the genomes and compare all of those annotations to a list of metabolic functions that has been curated by members of the Wrighton Lab as well as some of our collaborators. And so if we see a function or set of functions present, we'll say that this genome is capable of this set of functions. And then we can further distill those functional annotations to profile substrate utilization. And Kayla will go into this more detail later. So that first step is calling genes using Prodigal. Pretty easy process. We take our scaffold, so say this green bar is one scaffold from our bin, then we'll predict genes. So we'll draw all these little shapes on it that represent all the genes that are present on this scaffold. And Prodigal will give us that list of genes, their location on the scaffold, as well as their sequences in terms of nucleotides, as well as the predicted amino acid sequence. Then we'll take each of those amino acid sequences from each gene and search those across a variety of databases. DRAM does two different types of searches. The first type is a blast style search. So this is a direct sequence comparison of the amino acid sequence of your gene to the amino acid sequence of genes in databases. If you provide the KEGG genes database, we can't give it to you because it's you need to pay a subscription. But if you do subscribe to KEGG, we can compare to KEGG genes. You can also compare to UNIREF 90 if desired. This takes a very long time and a lot of memory if you're running it on your own. So we often recommend that people don't annotate against UNIREF 90. But if you have a sample that's relatively uncharacterized and you need to be able to get all possible annotations, the UNIREF 90 annotations can be very useful. We also compare to the MUROPS database. The MUROPS database is a database of peptidases. So this will tell you the ability of the genes in your genome to break down proteins and amino acids. Finally, you can search against your own custom database. You can give DRAM your database that is built. Your database that is you built yourself, and DRAM will search against that with a blast style search and return the best annotations from that. DRAM will also do HMM profile searches, so hidden Markov model profile searches against KOFAM if you don't provide KEGG genes, so you can still get KEGG orthology annotations. PFAM, it will search the DBCAN2 database, which is a database of HMMs that represent the genes that are in Casey, which Kayla will explain more in the coming slides. And finally, against VogDB, VogDB gives you information about the viruses that are present. So if there's viral genes present in your contigs or in your bins, this will be very helpful. So the raw level of DRAM contains all annotations for all genes in the database. This is just an example of what this table looks like. I don't expect you to be able to read this and really get anything out of it. It's just showing that we give you this big tab-separated file, which you can go through and gives you all the possible information you could want about all the genes in your genomes. Then the next level of summarization is the distillate. So the distillate is taking those raw annotations and summarizing them by function and telling you how many copies of genes with that function are present in your genomes. This tree here on the right gives you an idea of all the different categories that the DRAM distillate covers. So there's a very wide variety here, and we're always looking to work with collaborators to expand this to include more metabolisms that are potentially interesting to the wider community. And then the DRAM product is an interactive heat map of key metabolic functions by genome. You'll be able to use this in KBase and be able to go through each genome and understand the functions that each gene is capable of. And again, Kayla is going to further explain this in the coming slides. And so now I'll hand it over to her.
Okay, I'm going to share my screen, which is this. Share. Okay, and then can you see my slides or can you see the notes?
Can see your full PowerPoint
Cool.
Normal editing view.
Okay, cool. So let me just do this. Okay, so hello. My name is Kayla. Thanks for joining us today for this DRAM tutorial. Oops, I have to click. Okay. So specifically today, I'll be talking to you about the metabolism portion of DRAM and how to infer metabolic potential from DRAM outputs. And so to demonstrate this, we chose soil relevant genomes to showcase DRAM capabilities. And so we chose soil due to the diversity of metabolic potential harbored in soils. So this figure that I'm showing you is highlighting the important ecosystem processes that are carried out by soil microorganisms, including things like methanogenesis, methane oxidation, and denitrification. So we pooled several genomes for biochemically relevant processes, the ones I just mentioned, while also accounting for the divergent lineages. So for the rest of this DRAM lecture, we'll be using a set of genomes, a set of 15 genomes.
Kayla?
I'm not sure we're seeing your slides correctly.
Okay.
I'm still seeing Mike's ending slide.
Oh, so it's like not advancing.
Well, it's not showing screen mode either. It's just showing like in the regular PowerPoint edit mode.
Try stopping and restarting.
Okay, so should I just, okay, let me stop share. You stop share. Okay. Share screen. Maybe if I share my desktop and not the PowerPoint, that will be better.
Okay. Yeah, now I'm seeing your desktop.
Okay. And so can you see the slides advancing?
Yeah, now it seems to be going.
Okay, cool. Okay, so I'm gonna restart. Okay, so I'll just start here. So the point of the slides I'm going to present today is really to showcase the metabolism that is outlined in DRAM. So as Mike mentioned, beyond annotation, DRAM really does distill metabolism into functions that are biogeochemically relevant. And so for the rest of this lecture, I'm going to be talking about genomes that are soil genomes, but that really showcase the breadth of metabolism that DRAM profiles. Okay, so we'll start with 15 genomes from isolates and metagenomes, and we ran them through the DRAM annotation to obtain annotations for each gene within each genome from the 6 different databases that DRAM profiles. From there, we ran the distillation step, and so I would like to note here that DRAM outputs a large number of files, and all of them are linked. So you can start with the raw and wheel your way all the way down to the product and really get down to the gene level, whereas the raw really gives you all the annotations for every gene. And so we're going to start today with the product because this provides you with the most succinct level of metabolic summary of your genomes or mags. And then the distillate is one level up, and then the raw. Okay, so today we're going to start with the product. Okay. So so what you're looking at here is the actual heat map product. So this is the output of DRAM. This is exactly what DRAM gives you. And so each part, you can see that the product is 3 different sections. So there's a pathway completion, a subunit completion, and then a presence absence portion. And so each part of this heat map gives different estimations on completion of certain pathways. So for pathway completion, this considers alternate genes and for the pathways. So what you're looking at here is glycolysis. And so you can see that there, each little circle here represents a different KO or EC number, so a different annotation for this particular function from glucose to glucose 6P. And so DRAM really calculates DRAM calculates all routes and then reports the coverage for the path with the largest completion number. So here I'm showing you 2 different routes. One is 100% complete and one is 89% complete. Both are considering the alternate routes. The subunit completion, so this is really focused on the electron transport chain. So DRAM considers the number of subunits required for a given complex and then reports the completion for the subunits. So here I'm showing you NADHD hydrogenase, and so what you can see is that for one genome, this NADHD hydrogenase is 100% complete, so it has all of the subunits for this enzyme, and then for another genome, it is not complete, and it in fact only has 3 subunits. This becomes really important because having specific subunits annotated as NADH dehydrogenase could be indicative that this is in fact not an NADH dehydrogenase, but a trimeric hydrogenase. Okay. For the presence absence of specific functions, some require the presence of a single gene, while others require one or more annotations from sets of genes to be present. So for example, if we look at methanogenesis, we're requiring an MCRA here. Okay, so now that we know a little bit about how DRAM compiles the genome annotations and profiles specific metabolisms, we can start going through these genomes. So again, the genomes are on the left, and then the different pathways or functions are across the top. So two things I would like to note quickly about this heat map. It is interactive, as Mike mentioned, so when you download these HTML files from KBase, or you run DRAM on your server and you download the HTML files, you'll be able to mouse over and see how we are calling these genes. So for example, if you mouse over, so I took a screenshot here. This is a screenshot of what it looks like when you are scrolling across the heat map in the interactive HTML. And so you can see that dichloromonas, if we mouse over denitrification, so the reduction of nitrate, you can see that this dichloromonas does have a nitrate reductase, and that's how we're calling this gene. The second thing to note about these heat maps is that they are scalable to thousands of genomes. So you can scan your genomes quickly for particular metabolisms of interest. So if you're really interested in photosynthesis, you could go through 1,000 genomes pretty quickly and figure out which genomes have that function. Okay, so when first working through a genome or sets of genomes, we're gonna go into, okay, I have this heat map, now what do I do with it? When we're gonna start going through these, we're first interested in is how are these organisms potentially making a living? So our first question is, what types of energy generation metabolisms are encoded in my genomes? So the first key way for microbes to make energy is through respiration, and we can see this really easily in the DRAM product output. So in that DRAM product output, we would look for things like a full electron transport chain. So you remember we have the subunit completion there, a near complete TCA cycle. So this would be in the pathway part, as well as the potentially used external electron acceptors. So things like nitrate or iron. And so this would be indicative that your genome can respire and thus is making ATP via oxidative phosphorylation. The second way that a genome or an organism can make energy is through fermentation. And so this would be an absence of those pieces that I just mentioned. So you might not have an NADH dehydrogenase, your TCA cycle would be incomplete, and you'd be lacking those external electron acceptors. And so that's what makes DRAM really nice, because you can quickly look at this product heat map and see, okay, where do my genomes fall in this fermentation respiration? And so what I'm showing you here is the product output of those soil genomes. And so if we go and we look at the TCA cycle, we can see that we have some that do have a complete TCA cycle, so at 100%, this dark blue color. And then we can go over to our electron transport chain, and you can see that we do have some with complete NADH dehydrogenases, as well as other parts of this electron transport chain. And so from this, we can really start going through our genomes and saying, okay, dichloromonas is a respirer, disulfovibrio, yes. And so what we see here from these 15 soil genomes is that 10 of the 15 genomes have the potential to respire in their genomes. So our next question might be, of the organisms that can respire, what is their energy source? And so this is also showcased in that DRAM product output. Again, this is the product heat map, and you can see that the oxygen is housed here. So you can look at these, is oxygen the external, or the electron acceptor, is nitrate, is sulfate. We can also look for photosynthesis. So we can see that we do have a photosynthesizer. This green indicates that this Synecdococcus or cystis is a photosynthesizer. We can glean that disulfovibrio can reduce sulfate. Same for dichloromonas, can reduce nitrate to nitrite. And this is all in that output. You might be interested in other metabolisms, things like methanogenesis, and we do see we have a methanogen in this data set. So methanosarcina is present, and we know that because there is a functional MCRA gene, so the gene that's indicative of methanogenesis. And then we can also go further and see which substrates of methanogenesis this methanosarcina is using. So for this one, it can use acetate, trimethylamine, dimethylamine, and methanol. Okay. So we can now get some sort of idea of what, how our genomes might be making a living in these soils. But we might also want to know what kind of carbon they are using. So organisms also need carbon, and so we can get this information from the DRAM product output as well. So we have short chain fatty acids profiled, so things like acetate, butyrate, and propionate. And so for these, we do want to be careful because some of these enzymes can go either way, so they can produce it or consume it. But this does profile those genes for you. The other thing that it profiles is the Kzymes. And these calls in the DRAM product output allow users to profile the carbohydrate substrate utilization profiles for their input genomes. So for things like big carbon, things like pectin and starch. And I do want to pause a minute on these Kzymes because you might be asking yourself, what are Kzymes? These are enzymes that degrade or modify glycosidic bonds. And so these are for large carbon polymers. And so I have a few pictures of what those look like. So beta-glucan, xyloglucan. And to annotate these Kzymes, we use dbcan2. So in DRAM, DRAM annotates all the genomes and all the genes using dbcan2. And this gives you, what this gives you is a family level information for potential hits. So you might put your genome in there and get that you have 10 GH12s, 10 GH11s, and you get this sort of carboglycoside hydrolase profile in your genomes, but it doesn't really tell you anything about the potential substrates that they're utilizing. And so what DRAM does, we teamed up with Phil Pope and his team to really curate this database. And so what this means is that we were able to classify the dbcan calls that were to like a GH4 into things like pectin. And this expert curation and logic is housed in DRAM. And so this is a really nice feature of DRAM because it allows you to link those GHs to relevant substrates. And so for the DRAM product, this is what I'm showing you here. So this is that interactive heat map. The genomes are on the side, and then the different substrates are along the bottom. And so what we required for this to be true, so for a green square instead of a blue square, that your genome, this cytophaga, can degrade polyphenolics. We required that you have both an endocleaving and an exocleaving Kzyme, and so this was really to reduce false positives. And so this potential that is DRAM really allows the user to tie what a genome can do to, DRAM really allows the Kzymes to be functionally classified into substrates. Okay, so if we start breaking this down, you want to start to get to your overall genome metabolism. So what I'm showing you here is I pulled that dichloromonas genome, so it was highlighted throughout the entire liquor, product. And so it was highlighted throughout the whole way. And so this is the dichloromonas genome. And so what we're showing here is everything that's in the product output. And so you can see here's the electron transport chain. This is central carbon. You can start to see some of these electron acceptors. If we scale up a level, we can go to the distillate. So Mike mentioned this. And so this has things that are not housed in that DRAM product. So things like transporters, we can start to look at all the transporters that are in this dichloromonas genome. We can also look at the peptidases. So these things in green here, we have endopeptidases, dipeptidases. And so those we can add onto our, we can layer onto our genome metabolism to really understand what these organisms might be doing. And then the last thing is we can scale up even further and go into the entire genome and see what every gene does in that genome and start to really overlay other things that might not be profiled in the distillate. And all this information is available in all of the outputs. And so this is things like multi-heme C-type cytochrome. So DRAM does, it counts the number of hemes in every gene or hemotypes. And so you can start to pull some of those out. You can also look for other metabolisms that might be interesting in a particular genome. So things like PHB production and perchlorate reduction. And so with that, I would like to thank everybody who's had a hand in this DRAM annotation tool and distillation tool. And this is a big collaboration with the Sullivan Lab at OSU, as well as Simon Rue and Phil Pope. And it is available in KBase. I do want to draw your attention to the bioRxiv article, as well as the GitHub and the wiki. I do also want to highlight that much of the background information on the distillation product is available in that bioRxiv supplementary information. With that, I'll take any questions.
Thanks, Kayla. That was awesome. And Mike also. And we've got a ton of questions. In fact, there's been a huge amount of discussion going on over in Discord, which has been fun to watch. A lot of your colleagues are already answering a number of those, but I will try to pull up some of the questions that I think may not have been addressed fully yet. So there's one right here in the Zoom that says, does DRAM include heavy metal metabolisms?
Yes, it does. So I actually did pull that out of the heat map, but I don't think I have one, but it does. It has things like arsenic and mercury, some of those other electron acceptors.
Okay. Let's see, what other questions? So there was a lot of questions about coverage and whether coverage information is used in terms of quality.
Well, coverage, oh, that's a mic.
Yeah, I can address that. So coverage information is normally not used when assessing the quality of bins. As discussed this morning, coverage is heavily used during the binning process. So for example, in the narratives we went through this morning with some of the students, we use MetaBat2. And so MetaBat2 will take the coverage information along with the genome content and use that coverage and use those things together to be able to give you the best quality bins that it can. What these bin quality assessment tools, including CheckM, are doing are really just using the genome information from the bins to assess quality or completeness and contamination. So coverage information doesn't come into it at that level.
Okay. There were a number of questions also about integrating with other data systems. So I think you mentioned that DRAM already interrogates a number of different databases. So if I can pull up one of the questions, one question was if we do our own KEGG annotation, can we add it into DRAM before the files for step 2 or 3 are made?
So yes, well, sorry, no. Currently, DRAM operates on its set of databases, or you can add additional databases that you want to show up in your raw. So you can provide a database of genes as amino acid sequences, and DRAM will tell you if you have any hits against that custom database. We currently don't have any integration of custom HMM sets, although it's something we're looking into. And we don't have integration of custom databases at all into the distillate or liquor. We want to have this in here. It's just a little more complicated because in the distillate and the liquor, you have to define your own sets of genes. So essentially give a list of which gene or which database identifiers really are associated with each function, it takes some wrangling to make a pretty version of that. And so that would also make it very difficult to add custom databases personally. We're happy to work with people who want to add their own metabolisms to DRAM. especially if those metabolisms can be defined by identifiers that we already use, such as those from PFAM or KEGG. If we could use custom sets of those that are defined by the person who's an expert in this metabolism that they want to add, we're really excited to work with people to do that. And we also have a Google form that's linked to from the DRAM wiki and from the DRAM preprint that gives you the basic information you need to be able to have everything you want to add something to DRAM.
Great. Great. So one user asked a question, is this also including plasmid DNA?
We don't do any plasmid detection right now. It's something that's also we've looked at and is on our list, but it's not a super high priority right now. There's some cool tools that have come out recently for plasmid identification in the way that DRAM works. If you're annotating anything with DRAM, you give it a list of FASTA files. So a FASTA file is a file that includes genome sequences. And DRAM will annotate that with any hits that it gets from its database. What you could do is identify plasmids using one of these cool tools on your own, and then pass the FASTAs that come out of those tools into DRAM, and then DRAM will assign whatever metabolism or other categories that are included in the DRAM database and assign those to your plasmids.
Okay. And so some of these questions are also getting answered over there, but people were asking also about viral information and viruses.
Yeah. So we have a totally a related version of DRAM called DRAM-V, which we also talk about in the DRAM manuscript, which is DRAM for viruses. So DRAM-V specifically takes input from VirSorter. So right now we're dependent on you running VirSorter to figure out which of your contigs are viral. Then DRAM will take in the contigs that came out of VirSorter as well as one of the annotation files that comes out of VirSorter. And it will do its full annotation just like it does from MAGs, but then it does even more. We were really interested with this in identifying AMGs, or auxiliary metabolic genes. So these are genes that are in viral genomes that can affect the metabolism of their host. So these are really cool because they could totally change what your bin is doing in the community based on adding one or two functions that really came from the infecting phage. So what we do is we take all the annotations that come from DRAM of the viruses, then we look for we first assign how confident we are that that gene is viral, because even with tools like VirSorter, viruses have messy ends, especially if they're prophages. So we want to be sure that we're telling you a gene is really viral. Then we assign a series of flags which indicate our confidence that this is a metabolic gene. So if we have a gene that's both confidently metabolic and confidently viral, then we list these as potential AMGs. And we give you a similar distillate and liquor to what DRAM is doing based on the auxiliary metabolic genes that are present in viruses.
Okay, great. Here's another question that just came up. Does DRAM annotate origins/termini of replication either for chromosomes or plasmids?
No, we don't. So right now, DRAM annotates everything that is predicted by protocol as an open reading frame. And we also do searches for tRNAs using tRNA scan and rRNA genes using BARNAP. But currently we do not look for other genomic features. We're looking at integrating RFAM relatively soon so we can have some non-coding RNAs in there. But currently we're not looking for other genomic features.
Okay. So one other question that came up a lot, and it was already answered in the chat, but I'll give you a chance to give a little plug, and that is DRAM available in Keybase?
DRAM is available in Keybase as a beta app. So this means if you do the normal search of the catalog, like Alicia showed earlier, you won't see it. But there's a spot you can click for versions. And under that tab, there's something called beta. So you can click beta in there, and then you can search for DRAM, and you'll find DRAM in there. DRAM for Keybase is able to do the full DRAM for genomes workflow, and it will do this either on bins or assemblies. And you will get the DRAM output. And the students who will be working through the narratives with us this afternoon will be able to see that in action. And then everyone else will be able to see those narratives, giving examples of how to do it later today.
Great. Excellent. Well, there's a ton of discussion going on right now over in Discord, and there was a number of other questions in the Zoom chat. So I think Mike and Kayla, you guys are going to have your hands full looking through all the questions, but you've generated a huge amount of interest and excitement. So thank you very much, both of you, for your presentation. And we're getting to the point now where we're about ready to go back into our hands-on sessions with the students. And so those who are online for the public session will be coming back at 3:50 Pacific time. That would be about, let's see, that would be about 2 hours and 20 minutes from right now. So it's 1:30 right now here, and we will come back online at 3:50. In the meantime, the student teams will use their other Zoom links to go out to the break. And we look forward to having you come back. Those of you who are on the public session, we'll have some really interesting presentations this afternoon, including a talk about the capabilities of the Joint Genome Institute, and then a nice science presentation on connecting genes to function. So we really encourage you to come back and join us again. Thank you again to our speakers and to all who have participated. And we'll break now and go into our private session.